to tell you guys a story today, as I'm very privileged to have one foot in the university door and another foot in my own business as an entrepreneur. So I do a lot of lecturing at the university and I interact a lot with young minds. But of course, today I don't want to give anybody a lecture. And I'd rather want to tell you guys a story. My origin story is not necessarily with cannabis itself, but I find the world of cannabis fascinating and its history fascinating as I was planning my talk for today and how I can fit my passion into this talk. Now, my passion, of course, lies with natural products and how we could use this in every aspect of human life. Hence, I find myself in animal cell culture, making cultured meat. I find myself in product development with food sciences. I find myself in the spaces of generating genetically modified organisms, metagenomics, big data, analytics. I have trained myself as a biotechnologist for many, many, many years. I'm going to tell you a bit more about myself at the end. So let's talk about one of the oldest cultivated crops in the entire history of humankind, and that is considered cannabis. An amazing history lies with this plant, starting its story in Central Asia, making its way to China. It was used with emperors, it was used in tombs to mummify, and it was used to cure many, many ailments, as we all know already. It went all the way down to Africa, with the conquistadors to South America, and of course it ended up in the US. And that's where another story started with cannabis, although it's been cultivated and used very successfully for many, many years through many, many cultures that have developed very interesting systems and cures with the plant. If we consider the anatomy of a cannabis plant, we look at the whole thing, from root to flower. And every aspect of this plant is usable. We normally focus on the flowers and particularly the medical components. There's also a lot of people looking at fiber. But we can use every aspect of it: leaves, roots, flowers, seeds, fiber, and more. And every different culture have developed their particular variety. It's been hybridized over many, many years into what people need. At the top, I write there's 60,000 uses. Now, of course, this isn't true, but this comes from a story, The Emperor Wears No Clothes, where he actually wants cannabis legalized because it has many more uses than its medicinal use. So I found that quite an interesting story, The Emperor Wears No Clothes, and 60,000 uses for one plant. One could also say that it reached many people because of all the chemical compounds that it makes. I see this plant as a chemical factory. Now, from my perspective, this chemical factory produces from fatty acids to fragrances. We've all heard about terpenes, and they've got many other uses besides human health. It also makes carbohydrates to very strong fibers, which the Vikings also used on their ships as rope. Now, with my passion for natural products, I needed a way to understand how I could distinguish using natural products or using chemical compounds these plants. And of course, something that you are all familiar with, the word species, varieties, cultivars, chemotypes and chemovars in particular excited me. What's the difference between all of these? It lies within taxonomic classification. If we start with species, we look at the very, very start of a kingdom. And then what happened over the years was cultivated varieties or cultivars were produced with all of their interesting benefits, very akin to broccoli. We have also done exactly the same thing with broccoli, Brussels sprout, cauliflower, which is all family of the same plant. Cannabis is the same. There are natural varieties. There's lots of arguments going on as to what is a variety, what is a species, sativa, indica, ruderalis. I'm not necessarily going to focus on all the terminologies, but what excited me was the chemical composition and that there are ways for us to classify even cannabis plants based on their chemical composition or the chemical compounds that they make. Now, we largely focus on cannabinoids, terpenes, to classify something into a chemotype, which will give you the ratios and the content and quantities of cannabinoids and terpenes. Often we ignore all the other interesting compounds that can all provide us with a fingerprint, or what I would call a chemical fingerprint of a specific, giving us what we call a chemovar, a variety that can be completely distinguished by its chemical profile, its unique chemical profile beyond cannabinoids and terpenes. So how can we exploit this in a crop production or an agricultural process? How could we use chemovars, the total chemical profile, fingerprint of a plant, as an outlook for industry and perhaps research? So just out of interest, I saw that there are 5 different types of chemotypes. Type 1 and 2 accumulates THC, which is the aspect always in question in the legal situations. There are 3 other types that completely devoid of THC. And I'd love to challenge government bodies with growth parameters being relaxed for such varieties, or such chemovars or chemotypes, because the whole plant can be used. There are many, many more metabolites, many more chemical compounds beyond THC, which seemingly the only thing that regulates this plant. We know a lot about the morphology of the different natural varieties. We also know a lot about the morphology and the content of the different cultivated variety. Now that we know which compounds we can use for a complete fingerprint, or complete chemical fingerprint, is this something that could apply going forward to growers, breeders, research and rights? Chemically identify beyond DNA, beyond genes, beyond the genome. We start looking at chemical compounds for fingerprinting of plants. Is something that we understand from an enzymatic perspective. And in biotechnology is pretty easy, not only using the genes, but also using environmental aspects. We can manipulate and we can understand cannabinoid, terpene, and flavonoid profiles and how to increase and decrease them, and which remains constant so that we can actually use this as a means of chemical fingerprinting of plants. Chemicals that we shouldn't ignore in the plant. So if we are to exploit the chemical compounds in agriculture and crop production, what potential exists for biotechnological collaborations? Of course, micropropagation, there's a lot of tissue culture specialists in the audience. Clonal propagation, what are the troubles with this type of propagation going forward, particularly if we start looking at DNA changes that are brought about? Can we circumvent that using a chemical profile of a plant, using that to DNA or chemically fingerprint it? I think it's possible. Genetic modification, genetic breeding using particular genes or particular marker genes. Of course, we need information, genetic information for the plants to be able to do this. There's a lot of app development. And out of interest here in my slideshow, GrowDoc designed an interesting app which could show potential or has the potential to show different types of infection. And is this something interesting that we can look forward to for the industry? A quick app development for rapid identification of problems within our crop production facility. We can even, with our chemical information, also understand what type of infections plants have. So I think with us looking at chemical compounds and natural products, they hold a lot of value and they hold a lot of answers as to the plant health, plant environment. And lastly, for a lot of R&D potential, what can we work with? What can we work on? Where is the research and development potential? So I'm going to leave the R&D potential for my very, very last slide, where I want to leave you with a few thoughts going forward. We can, of course, design unique environments and unique growing conditions to drive a particular wanted chemovar. If we know what we want in our products at the end, can we drive our plants to grow in a certain way to make a certain product or a certain profile before we make a product for the market? Much in the way that we look at circular bioeconomies of cities and people and places and biofuels that we hear a lot about, I look at the cannabis plant as a circular bioeconomy plant as well. So I equate it with circular bioeconomy because of all of its versatility and the diversity with what you can use it for, the whole plant, every single part of it. So I want to tell you a little bit about myself. It's my second last slide. I am affiliated with the University of Stellenbosch, but I don't work for the university. I work for myself. I'm a bio-entrepreneur. But I have access to spaces and places at the university, and I also supervise a lot of young minds with a lot of very interesting projects. And I think from an industry perspective, to be able to collaborate and to be able to make strategic investments into the university and helping young minds to grow and to maybe come work for you one day is not a bad investment to me. From my previous slide, sustainability is very important to me. So also with the university, I work with QS Merit, which works with a lot of global universities as well. And we mentor young minds all across the world on sustainability problems, real world sustainability problems where businesses from around the world bring a problem to the table and a group of young minds tackle that problem. And they come up with some very interesting ideas on how to tackle sustainability problems. There's a lot of cannabis industries also participating there, and perhaps something for you to look into. This was started in 2022. We look at every single aspect of sustainability all across the world. Beyond that, I am the founder and CEO of BioSupport, Pty Ltd, where we love to turn no idea into any idea. And we have fun, and we do a lot of exploratory R&D. We do a lot of consulting, education. We ignite scientific passion and nurture young scientific minds. What can I bring to the table? Which ideas do I want to leave you with as a matter of thought? One of my big research arms is working on microgreens, plants with 2 to 4 true leaves. Very tiny plants at the very beginning of their life cycle, their development cycle. These plants, or the stage of development, is known to have significant amounts of secondary metabolites or chemical compounds. An idea to look at growing good quality cannabis seeds, an idea to produce microgreens, and perhaps see whether there isn't significant chemical profiles in that for product development. Next idea, hairy root cultures, the roots and the root systems of a cannabis plant is very interesting. A hairy root system is a factory in itself for producing chemical compounds in a laboratory in a sterile environment and can be scaled up to industry. Of course, the next idea is genetic modification. Can we incorporate particular desired traits using genetic intervention into cannabis plants? Yes. Can we drive using huge metabolic pathways and genetic modification? Can we drive particular chemical production within the plant that way? And the last idea I want to leave with you is growth conditions, which is absolutely no secret as to how this can change and alter a plant. So a lot of the microgreen research I do, I drive chemical compound profiles using high heat, high light, and vice versa. Also an idea to drive particular chemical compounds to be produced in this chemical factory plant using very basic environmental stimulus. So I'm going to leave it there, finish with my talk, and the floor is open for any questions.
I just wanted to ask, you've got quite a extensive knowledge on the genetic side of things. What are your thoughts on the new triploid variants that are coming out where they are breeding that extra chromosome to make sure that it's more stable? What are your thoughts on?
Increasing ploidy has its benefits and not benefits, but it definitely increases the diversity and the stability with which genome can move forward.